familiar thing all over, and we started the recording. The first thing I wanted to say was brought some organic snacks in today that are all cured with conventional celery powder, and I'll get into that more. But if you'd like to try some, compare a conventional product versus an organic product, you're welcome to do so. And Erin, I can pass it to you for introduction.
Thank you, and welcome everyone. It's a pleasure to see everyone on this beautiful afternoon, and I'm thrilled to introduce Owen Washam, who's going to talk to us about the work that he's been doing for the past couple years on organic celery powder. So Owen has been with us at University of Madison and the Department of Plant Pathology since 2020, so came during COVID. But prior to coming to UW-Madison, he's originally from Salem, Ohio, moved to North Carolina in 2016, and was an undergrad at NC State at Raleigh. He got his BS in plant biology, as well as a BS in horticulture with a concentration in research and biotechnology. And his undergrad research experience included small fruits, hops, hydrangeas, pitcher plants, ornamentals, and controlled environments. So he had a really diverse experience before coming to UW-Madison. Also, I know it's really been into pathology, mushrooms. So really started at a really hard time for graduate students right at the start of COVID. But he really, even with those challenges, dived into research, doing some initial greenhouse work to set the stage for the work that he'd be doing in the field starting in 2021. He was attracted to come to the Department of Plant Pathology because of our very interdisciplinary and collaborative way of working here. So not only did he get to have aspects of plant pathology into his research, but also organic management and horticulture, and some collaborations with meat science as well as physiology and entomology. So not only did he do greenhouse trials, but field trials and laboratory analysis with Dr. Sindelar, who just walked in. So I'm really excited to have Owen present all the work you've done over the past couple of years and then talk about the direction.
Yeah, thank you, Erin, for that introduction. So as Erin mentioned, this is my Ecit Seminar, and the title of my talk today is Organic Celery Powder, a meat curing agent for organic processors. And before diving into the presentation, I wanted to present an outline just to give everybody an idea of where we're headed in the presentation. So we'll start with an overall introduction, where's the organic market at, and move into our research questions, and these research questions that arose, and then we'll move into our individual objectives and talk about those individually and those individual conclusions, and then we'll move into overall conclusions and future work for this study. And then finally, we'll have I'll acknowledge the sponsors, and I'll have time at the end for questions and comments. And so to start off, there has been increasing growing demand for organic products pretty much since the inception of the National Organic Program in the early 2000s. And in 2021, those organic sales in totaled 63 billion. And of that total, 90% of the value is food sales. So that total in 2021 for food sales was 57.5 billion. So this slide is just highlighting the majority of the organic sales. It's in food. So this is the direction we're moving. And the meat product categories in particular show great potential for growth and have shown growth over the past couple of years as well, continued growth. So the challenge with curing or with preserving organic meat products is it's really in the organic rules. Typically, when you have an organic product, it has to be made of organic ingredients. And there have been some challenges in developing an organic curing powder for organic products, an organic nitrate source. So today, potassium nitrate and sodium nitrate that are used in conventional curing processes, and these elements have been used for hundreds of years in their basic forms. They're not allowed in organic. So these synthetic ingredients can't go across on an organic meat product. However, since the organic curing powders have not been developed and not been released, there is a special exception for conventional curing powders to be used in organic products. And that's where it gets a little bit confusing. Because that special permission, due to technological barriers and lack of commercial release, that expires in 2026. So essentially what this slide is saying is organic meat processors, they're facing a risk as time goes on here. So it's an imminent challenge, and we need to address it before that sunset date. And just highlighted here on the bottom, these are the organic meat snacks that I brought in today. But all the products, all the meat products are cured with conventional celery powder, even though the organic product is labeled organic. That's the exception that I refer to. But this is the labeled organic product, and Applegate is one of our sponsors. So moving into celery juice as an organic source. So celery in general, it's a known nitrate accumulator among other families of plants. And high nitrate is normally considered like a bad attribute. There are some health concerns with consuming nitrate at elevated levels. So the celery that you would normally find in the store, what we call table celery, would have lower levels of nitrate. They're not going to be high enough that it would cause a food safety concern. However, in this situation, we want high nitrate, and this is a processed vegetable you never see in the store. So it would be, going for a niche purpose. And nitrates from vegetables are bacterially converted or fermented through a chemical reaction from nitrate to nitrite. And the nitrates are used for organic meat curing. But here we'll be talking about the celery juice, which is the nitrate concentration. And the curing powders for celery, they've been shown to conserve traditional meat sensory attributes that consumers care about. These include smell, taste, color, and the curing powders that are produced. They must meet these sensory attributes so that they will be accepted by consumers. And celery in particular was chosen because of its low contribution of these sensory factors. It has a low color contribution. It has a positive sensory attribute. Celery is a savory vegetable, so that's kind of a plus. And other vegetables that we provide negatively impact these sensory effects. So that's why we went with celery and celery juice. There are many factors, like a bunch of factors that affects nitrate accumulation in plants. So nitrates are accumulated into the vacuole, the water storing cells within plants. And some of these factors include fertility management, type of fertilizer, whether it's organic, whether it's synthetic, when it's applied, whether it's pre-plant or within season, plant species and variety, even within plant species. We've seen from previous research that different celery varieties can be widely variable based on a varietal level. Light intensity is another factor. So nitrogen reductase drives that nitrate to nitrite conversion, and it's light activated and driven. So during the day, you see typically these nitrate levels drop within the field by a certain percent, and then they rise again overnight during the dark periods. And then finally, crop maturity. It's been shown that older tissues accumulate more nitrate than younger tissues. And this is hypothesized because as younger tissues are growing, they're putting that nitrate, they're accumulating into biomass. So they're growing, and they're putting this nitrate into growing bigger. Whereas crops grow, as they approach maturity, that nitrate in excess can just be stored. They're not looking to put on more biomass at that point. They're plants, they don't look, but they are not accumulating biomass at that point. There are some effects of concern that have been raised about using a higher nitrogen fertility rate, and that includes increased pest pressure, and particularly in sucking insects. Two that I've exhibited here are the green peach aphid and aster leafhopper, which is the model organism that we chose. And those downstream effects from there can change plant health status. So essentially, these sucking insects are inoculated with, they harbor viruses or they're vectors for diseases. And then as they're feeding on new crops, that's a potential inoculation point from their feeding activity. So I've already highlighted some of the issues with organic celery for curing powder. It's tough to get consistent levels of nitrate within organic systems. The industry is asking us for a 1,200 part per million threshold. And there's a lot that goes into that, a lot of economics and a lot of things outside of my scope, but that's the threshold that we worked with. And the differences also arise from the different sources. As I mentioned, from conventional mineral fertilizers, those can be plant available immediately. You can see pretty much an immediate response. But within organic systems that require organic amendments, such as manures, green manures, such as cover crops, those nutrients aren't necessarily available and they're not necessarily available immediately to the plant. So they have to be mineralized within the soil before they can be taken up from the plant. And that's what's unknown. That's what we're trying to tease out, is what is this curve over time within a celery season? There are concerns from the National Organic Standards Board, the NOSB, that over applying nitrogen, as is done in conventional plots to get this high nitrate, they're concerned about that because there are definitely negative environmental impacts on over applying nitrogen. And part of the organic standards and the goals of admission of organic management is really conserving that soil, protecting it, and really being judicious about applying nutrients and being stewards towards the soil. So there's a need to balance the nitrogen rates in order to meet the economic threshold, so that the industry threshold, but not cause environmental impact. So we're trying to tease out, what's the minimum we can apply per se without causing an environmental impact? What is the rate that we can apply without causing an environmental impact? Production of conventional celery powders. As I mentioned, it's usually mineral fertilizer driven. And this industry within the organic curing powder, it's closed and it can feel proprietary when trying to request information. So it's difficult to get these details that we know will affect the nitrate concentration from industry. And as I mentioned, the higher nitrogen fertility applications are used to produce that raw celery product for curing just by putting more and more nitrogen on. And another factor is, so within the organic system, this celery is used as a processing crop. So as compared to like a cash crop that would persist, throughout the whole season, you'd earn more on that cash crop than you would this processing crop, just by rule of thumb. So one of the questions that we had is how do we use this limited information to about growing conventional celery for the production of curing powders to meet the regulatory goals, regulatory requirements and the goals of industry. Here are some of the needs that are highlighted to produce the organic powder. What are the best management practices within an organic system to produce high nitrate with no intended environmental consequences? We looked at variety selection and looked at fertility sources. We used 2 different types of organic fertilizer, green manure, and then a organic chicken derived manure. And we looked at a range of fertility rates from a low rate to the middle rate, the state recommendation for table celery, and then a high rate as well. And then finally, the variability across regions. We did a one-site study, so we didn't see a lot of variability within our study, but we've seen in preliminary data that these nitrate concentrations can vary across locations, soil types, organic amendments. Does managing we have a couple questions that arise from these, and we want to confirm, does higher nitrogen, how does it impact the pest pressure? Does the organic curing powder differ from its conventional counterpart as far as food quality attributes? And what are the economic constraints related to the production of organic celery from seed all the way to the juice in the powder? So this, as Erin alluded to, this is a very interdisciplinary project, and we have partners from across the U.S., including here in Wisconsin and in Florida. we're working with the University of Florida doing replicated trials of what we're doing here, but that would kind of look at the different soil types and different environmental factors. And we're also working with industry, Kerry Foods here in Wisconsin, Diane Foods, and of course Applegate, the producer of the organic salami snacks here. And the focus within this presentation is the organic management of high nitrate within celery within Wisconsin. So although it's got a very big scope, we're just looking at Wisconsin here. And the results could be extended to the Midwest. So in summary, our research questions that arose from this was how will applying the pre-plant organic nitrogen fertilizer impact celery nitrate concentration? And going forward, I'll use cons for shorthand of concentration, so cons concentration. And then will high rates of N nitrogen result in greater aster leafhopper abundance? And will high rates of nitrogen negatively impact on health? And now I'll move into each of those objectives. So objective 1 is managing the organic celery for processing into a nitrate source. Our experimental design was a 2-year study at Madison Organic Agricultural Research Station on certified organic land, conducted between seasons of 2021 and 2022. And we used a split plot randomized complete block design with 10 treatments replicated 4 times. And our whole plot factor was the peak cover crop, and that had 2 levels, with or without. Our split plot factor was the nitrogen fertilizer rate from a range of 0 kilograms per hectare all the way up to 336.25 kilograms per hectare. And just for ease of this presentation, I'll abbreviate the treatments as N0 through N336, illustrating just the high and low range across the board of nitrogen rates. This is a list of just kind of important dates and important agronomic dates when we're trying to tease out these effects. When we started with the celery, started from seed, organic certified seed, and worked with a partner greenhouse in Viroqua to seed the tall Utah cultivar celery in the greenhouse on March 10th and March 18th of 2021, 2022, respectively. We chose tall Utah because of its performance in preliminary work before I arrived in the lab. It performed well on the basis of nitrate accumulation and resistance to diseases, work done by Dylan Bruce. And then our green cover crop, green manure, was planted in April of 2021, 2022. And the field workup is detailed here. Essentially, we terminated the cover crop, hand broadcasted the pre-weighed organic nitrogen fertilizers, and then shallow tilled at 15 centimeters or 7 inches across the whole plot before transplanting celery. So that's what I mean by pre-plant. All the fertilizer was in before. And the celery field transplant occurred very soon after that application. And I'm going to use days after planting to mean days after planting, days after fertilization interchangeably. This slide is essentially highlighting that we're looking at these dates across years as equivalent because they were timed by days after planting and fertilizer applications. And this just further illustrates that, and I included some pictures here. This was a picture, it's kind of small, but hopefully the Zoom can see it. This is at planting. The celery seedlings are about this big. They're in 72 cell trays, and then this is at 30 days. Increasing biomass to 60 days. And then finally, this picture was taken right before final harvest at 90 days. And so these harvests that we did every 30 days, we're looking at nitrate concentration. We want to measure them without taking the whole plant because we want to measure it over time. So we're removing petioles, a standard amount of petioles each harvest at 30 days, 60 days, and 90 days. And the nitrate was measured at each plot. So within each replication, we also tested the variance within replication. Moving into the nitrate monitoring and analysis, after we harvested petioles, within 24 hours, they were processed. And that's to cut down that nitrate conversion to nitrate. But focusing on our slide here, essentially the celery was juiced with a commercial juicer, as you see here, diluted from 1:1 to 1:16 to be within the range of the refractometry device that we use to analyze the nitrate. And we pipetted 7.6 microliters of diluted celery juice solution onto each test strip, and took 3 readings per plot, per sample, to average those to get our parts per million numbers. And I just wrote analysis consistency is key with refractometry. There's a color change over time with the test strips. So that was ensured, whether it was myself or working with undergrads. It was a very, it was a very, I feel like it was a very consistent process. The last bullet point here, the bulk juice from the celery pustules was always stored frozen. As soon as it was juiced, it immediately went in the freezer. Whenever we needed samples for analysis, we would thaw smaller aliquots of those frozen celery samples, and then centrifuge them and dilute them in preparation for analysis. And those samples were kept fresh and cool. And what I mean by fresh is if we had a diluted sample that was over a month old, it was out, it was in the trash. Make a new one. That's why we have the bulk juice in the freezer. And the freezer helps preserve those nitrates from degrading into the nitrate. So moving into our results here, over our 2-year study, we found that nitrogen rate is essentially causing a positive, it's a positive relationship with the nitrate concentration within the celery. As you move down here through the nitrogen treatments on the left, from 0 to the high rate at 336, you can see the means here at 30, 60, and 90 days after planting are increasing over increased nitrogen treatment. When we did statistical analysis on these and separated the means, we found in 2021, we found 2 groups that were significantly different. And in 2020, I'm sorry, that was 2021. And in 2022, we found 3 groups, A, B, and C. And each one of these groups is indicated by a small letter here, A and B, and that will be highlighted on the next slide as well. But essentially what all these numbers are saying is that applying over the state recommended rate of nitrogen of the 134 treatment is likely required to produce these industry requirements of nitrate concentration. And these means for the groups 201 and higher were significantly different than the 2 treatments in the lower group. And this is within the green boxes here, you see the data that I just presented just in a different way. And this is just highlighting the stark differences between our higher nitrogen rates here in the green box. This is the same green box you saw in the last table, and our lower nitrogen rates here of 0 and 135 nitrogen. These are significantly different, and they wouldn't likely meet industry requirements of 1,200 parts per million. And one thing I do need to mention is that though we saw a positive relationship with the nitrogen rate, within the green boxes, these means are not significantly different. So essentially what that means is, we're getting statistically the same result if we apply the highest range of nitrogen versus the 202 range of nitrogen, rate of nitrogen. So there's really no need to apply that above that rate, because you likely, based on this data, wouldn't experience any additional benefits from that extra application. Looking at our conclusions, so although our highest nitrogen concentrations were measured at the earliest date of 30 days after planting, the lower harvestable biomass at 30 days would not justify an earlier harvest. And that's what I highlighted in the pictures, just showing the growth over the season. When the celery is at 30 days, it's really establishing in the field at that point. It's this high off the ground, and you can hardly walk through the fields at harvest. They're pretty tight at that point. And processing crops, as I mentioned, are relatively lower in value compared to cash crops. So the farmer has to make a decision between, I have one season. Should I plant a cash crop, a process crop? The cash crop is the way to go if you're looking at the pure dollar amount, because in general, the processing crop is lower value. However, shorter season celery season may be warranted. Like if we could do a double planting within one season or follow the celery with a cash crop, If we could harvest those earlier on and gain more nitrate that way, that might be warranted for this specific purpose. And also, just due to the variation within organic on organic amendments, soil types, environmental effects, we need to do more Midwest studies to study curing powders to investigate how nitrogen fertility impacts nitrate concentrations across locations and management systems. Moving into objective 2, just a reminder, we're looking at nitrogen fertility impacts on organic celery, crop health, and then also biomass. So fresh harvest biomass, this was a different harvest than our nitrate monitoring harvest. This is when we took pictures of every plot and removed the plants here. You can see at crown level, and that's essentially at the soil level. We're taking a harvest knife and cutting it down at soil level, harvesting into bins here and measuring them at a kilogram rate and converting that rate into kilograms per hectare based on the area of these highlighted data plants here. And when we were harvesting, for all of our studies, we looked at these 12 middle plants in rows 2 and 3 and considered those our data plants, whereas the outside plants we considered border rows. So those were not measured for nitrate or response variables. We also looked at crop health and aster leafhopper monitoring. So as I mentioned, sucking insects are an issue with especially disease vectoring, and aster leafhopper is a known vector of diseases within apiaceous crops such as celery, carrots, and we wanted to use that as our model organism. So for plant health rating, I had trouble finding data out there for processed celery, because again, the consumer attributes aren't so much as bad, aren't so much a concern when it's going into a juice and then that juice is being converted into another product. So I had trouble finding those. So I used conventional celery grading literature and data out of California just to help guide my development of like a field rating and field key. And for each observation, we did observations from June to August in each season as weather allowed. And that meant 5 weeks of aster leafhopper counts and 7 weeks of plant health ratings. And as I mentioned, the aster leafhopper, we used it as a model organism within this crop. Getting into our materials and methods, first for aster leafhopper collection and counts, what we did was for each plot, we had repeated consistent net sweep collections. So that's myself and a student helper going out to the fields and sweeping a standard amount of sweeps, 12 sweeps or 6 per row across the plots. At that point, we would essentially, I would say, wrangle the net so we could put it in a bag, a big clear Ziploc bag. And this is on recommendation from Russ Groves, who I have to thank for this protocol. And we put it in the freezer, and that essentially killed the insects off, and so we could positively identify them at that point. And we did that across the plots, across the weeks. And then the bag readers, so these were, again, myself or undergraduate helpers, they were trained on identifying the aster leafhopper, which is characteristic by these 6 lines on the face of the insect. There are other sucking insects like other leafhoppers that affect celery, but we didn't look at those. For the crop health rating scale and field key, as I mentioned, I had trouble finding this, so it was one that we sort of developed based on observation, and I used the USDA grading for table celery, and again, that just highlighted the difference between celery and processed celery. I looked at it on kind of a sliding scale based on a paper that I used on measuring plant disease, and essentially as severity increased, I rated a higher rating. And so you can see here on the right with a 5 rating, this would be considered like a dead plant. It's not coming back from that rot that happened through the crown. And then 0 is a completely healthy plant. And then 1 is, spotted incidence, and incidence is a count based on the population, whereas severity is the progression. And so we looked at the 1 and the 2 ratings were lower incidence, whereas the 3 through 5 ratings, it was more of a systemic issue. I started to see, more signs and symptoms among more plants within the plot. And again, we use the same data plants for rating. Moving into our results section. So within 2022, we did see a slight positive relationship between nitrogen treatment and biomass. We didn't observe this in 2021. And you can see that why I say marginally significant is because our p-value down here for biomass here in between yields is 0.008, and our alpha level was set at 0.1. So by that definition, this is a significant result. And looking at the harvest between years, I suspect there's the biomass difference that you're seeing between years here is likely related to climate. And the next slide that I'll present here really just shows the stark difference. On the left, you see both of these are the same treatment. And on the left, you see a first replication of a nitrogen 202 treatment. And this is on August 31st, 2021, similar to what we see in an identical replication prior to harvest in 2022. And you can see just, the visual density, and these plot pictures exist on these dates for all 40 plots across the trial. So clicking, moving through those, that is a consistent observation that I made. So nitrogen fertilizer rate, we didn't find an impact on aster leafhopper population. And as you look here on the left, on your right, we have 2021 and 2022 data. And you can see in 2021 just the stark differences in collections. And week 4, we had almost 30 insects, but in 2022, we didn't have more than 7 insects collected at any given time. So we saw a significant year-over-year result, and I'll get into why that might be happening in a minute. And weekly counts within years. So we tested within years as well, and we compared, July 1st to 13 to 21. We found that they were significantly different, and those p-values are presented there. And we're basically the conclusion that we're drawing is the climate impacted aster leafhopper more than nitrogen rate. In 2021, the weather patterns that normally deliver aster leafhoppers from the southern U.S., it was a very conducive environment for aster leafhopper breakout and development. Whereas in 2022, we had one major storm event in the spring, and that that very well could explain why we didn't see. There were a lot of weeks where we had 0 observations. And talking with Russ, this is what we went over, that those weather patterns that normally deliver aster leafhoppers just didn't happen in 2022. So that kind of explains that big difference. With our celery plant health rating, this data, it was more observational. As I mentioned, I developed a kind of a novel key and rating scale. So it wasn't analyzed based on that and based on the data not fitting the assumptions of ANOVA. So here I just presented the raw data presented over time over the 7 weeks. And what these trends are showing in 2021, when I mentioned the weather patterns, mainly precipitation and storms, were occurring, we see an increase in disease pressure here. And we experienced a lot of losses in 2021, almost 50%. And this was due to rot and water contact, whereas in 2022, we didn't experience any losses at all. Every celery count we cut into it looked great. So those numbers are different on that basis. And it's important with plant health rating and organic to do early scouting and proactive roguing. And what I mean by that is there were weeks where I collected no data essentially, but I was out there observing the crop before when we would expect diseases and pests to arrive, in order to get just kind of a starting point. We're seeing very healthy plants right now, and then that starts the observations that change over time. And the severity, as I mentioned, it was greater in 2021, but in short, limited in 2022. Moving along here, we have about 6 slides left. So conclusions from objective 2, just a reminder, we explored how nitrogen fertility management affects organic celery yield and crop health and aster leaf hopper populations. Irregular precipitation events that I mentioned in 2021 likely impacted these responses when compared to 2022, greater than our response of any nitrogen response that we could measure. Our study did not observe a relationship between nitrogen fertilizer rate and aster leaf hopper populations, though other studies have reported this phenomenon. And results on these studies can differ based on the management system, and that's kind of outside of my scope, but between organic and conventional, we need to have more comparison studies because those results differ on management system. So overall conclusions, getting towards the end here, the takeaway from my project was the nitrate concentration is influenced by the amount of organic nitrogen applied prior to celery planting. The higher nitrogen application rates are likely required as compared to fresh market production for this purpose. And as of now, growers should continue to follow the state recommendation for celery production and avoid unnecessary costs and detrimental environmental impacts that may arise from over-application of nitrogen. These experiments conducted identified factors important to pest populations and celery diseases such as precipitation and climate. Those were some very strong impacts that we observed. So what's next? I mentioned this is a larger project. We have Jeff Silva here from the meat science department and his student, Stephen Sheng, my counterpart over there working on the meat curing side of things. The celery juice that we produced is currently in the lab and being converted to nitrite, and it will be used to cure hot dogs. And my counterpart in Florida, Zachary Ray here, I had the opportunity to meet him at a conference in August, which was awesome. And he's doing replicated trials as well over a longer period to study how nitrogen rates affect that nitrate concentration on a Florida soil and in a Florida environment. And finally, economic analyses and extension of results will be completed. The economic analysis just kind of looks at those costs and benefits of our recommended management practices starting from the seed all the way to the powder. And that's outside my scope, but Dr. Paul Mitchell over in the Applied Ag and Agriculture Economics Department is working on that. And then finally, extension of results. I was able to present at 3 different conferences last year, extending these results to peers. And it's been really great to have that experience while here at Wisconsin. Oh. Can't skip over the people who I need to thank most for the last couple years. So first off, I want to thank my family. My wife Jenny is here today. She has been so graceful to me and just been very understanding through all the stress and just the challenges that grad school, it just comes by. And then my parents, my father Scott is here today, has encouraged me all my life and just supported me from anywhere. Always available for a phone call, and it's just great to have him here. And my mom is zooming in over from Ohio. She has a foster child right now, so she can't travel. And my sisters are also there. Thank you to them. And I also want to thank the Silva team, many of you who are here today, most of you are here today, and past and present, Dylan Bruce that came before me, working on the celery, and of course the undergrads that we had over the years, and Stephen Sheng over the meat science department, and Zach Cray in Florida, and then of course our industry partners, Terry here in Wisconsin, Applegate, and the others that we work with. And then finally our plant pathology department. I came here and COVID kind of broke the expectation of a family environment, but boy, did we rebound. I feel so supported by my peers, and that's what got me through stats and the really hard part. And it's nice to be able to be oh, this is the problem that I'm having. I can empathize. That's really nice and something that I'll never take for granted again. So thank you, and at this point, I'll take questions and comments.
So, yep.
I was gonna,
Chuck Courtney?
so I'm just curious, when you got to meet the collaborator at UF, did he also see this crazy variability with like weather and stuff with his data as well? Or
Not at the
do you-
point when we discussed it.
Okay.
Yeah, he was seeing more of a nitrogen, a nitrate. So I'll repeat this real quick for the Zoom before I get into the answers. So the question was, did my counterpart in Florida observe those similar effects over time and location that we observed here in Wisconsin? And in short, no. The soil type in Florida is not a nitrogen holding. They have a lot less organic matter in their soil, more sandy type, and that's more conducive for that nitrate leaching. So as soon as that nitrate is available to plants, it might be already down the road. Whereas here in Wisconsin, on my field specifically, we had between 2% and 2% of organic matter or higher. So that was a good soil type for keeping this nitrate plant available. Francois?
Yeah.
Yeah.
And congrats. It was beautiful to see that presentation and to see like the results of 2 years of like commitment.
Yeah, thank you.
I was just a little curious between your recommendation like that the higher rate is likely needed, but you're saying that farmers should stick with the state recommendation. So what is your recommendation to farmers that you found out that there is likely a higher rate needed?
Yeah, that's a great question. So just to reiterate, Francois in the room here asked, I talked about two different rates, a state recommendation rate and a higher rate. And what would be my recommendation to farmers? And I'm going to skip a couple of slides back here to the nitrogen treatments and just kind of highlight. Okay, so when we originally started this experiment, we were working with Wisconsin Vegetable Growers Handbook, which is in pounds per acre. But for scientific studies, we go to the metric system. But for these rates, 0, but for 134 kilograms per hectare of nitrogen, that is the state recommendation. And converted to pounds per acre, that's 120. And the rate above that, the 201 kilograms per hectare is above the state recommendation, essentially. And what I would recommend to farmers is stick with it for now, because stick with the state recommendation. I need to be more clear. Because this was a 1-year study, and we really need to see results from on-farm trials across Wisconsin, across soil types. There's a lot of celery grown in muck soils, which we don't have here in Dane County, and I would expect different results from those. Does that answer your question?
Yeah. Thank you.
Oh, and we have a question here from Zoom. Dylan Bruce asks, do you think the agronomics are approachable enough that industry will be able to respond to the 2026 deadline?
Yeah, that's a great question, Dylan. Thanks for asking that. So the 2026 deadline has been extended based on, just for some background, technological barriers. It's been worked on for a long time, trying to get these nitrate rates from an organic system. And there has been a product released from that's organic celery powder, and it's been released for commercial use. But that's the only one out there that we know of by Diana Foods. But I think that they would be able to meet this maybe using other vegetables. So these could be, I know there are studies that will be happening after me that look at Swiss chard. And Swiss chard is another nitrate accumulator in the family Canopidaceae that preserves those meat quality attributes that I alluded to earlier. So maybe not with celery, but other vegetables are being explored. And those shorter season crops seem to be the way to go, because right now we really can't say to farmers, you know, it's justifiable to have a whole season dedicated to a processed crop when there are better economic options out there. You know, better is a loaded word, but better economic is key. Yeah, Rachel?
It was interesting with like the middle to high nitrogen concentrations. It looked like early on there were differences, but then they all kind of decreased to the same level. Do you have an idea like what's going on there?
Yeah, so Rachel asked here in the room, why are we seeing these differences across time within the nitrates? They look like they're in a similar group and then they drop. So this is kind of the agronomy side of the organic fertilizer. 30 days after planting, we're expecting the celery. The celery is accumulating nitrate from that nitrogen fertilizer that's being mineralized in the soil, and it's readily we predict that it would be readily available at that point at 30 days. And at that point, it would be putting into growth and have high nitrate. And then as that organic fertilizer would be depleted, the nitrate levels we hypothesize would drop and then rise again as crops approached maturity. So we didn't necessarily see the rise that we were expecting, but we did see that positive response, and the averages across the higher means were statistically similar. Did that answer your question?
I guess what I'm
Can I
curious
clarify?
I guess what I'm curious is why do they all decrease to the same level? Like, why aren't they parallel lines, I guess?
Yeah, are you asking more like
The
why
ones
aren't they
in the
like
green
this?
box.
Okay, the ones in the green box, why do they go straight across and like parallel instead of a decrease?
There seems to be like a limit.
Yeah, I'm really not sure. And that's one of the things we see in reviews looking at nitrate concentrations in celery, is like this is just the 2 years of data that I collected. And the range for organic celery in the review is like 1 ppm to 3,500, 3,500. So it's not necessarily something to say that it's like, yeah, I'm not sure why we're seeing that specific trend. Yeah,
Follow-up question to that.
Mhm.
So you applied a lot of nitrogen upfront at planting, right?
Mhm.
You're seeing this, especially under the higher nitrogen rates, the 3 higher nitrogen rates.
Mhm.
You're seeing a
box.
very high level of nitrate in the beginning of the season. Well, relatively speaking, right? 30 days after planting, it's high,
Mhm.
and then we're all going down.
Mhm.
Everything over the course of the season to the end of like 90 days after planting. Where do those nitrogen go?
Where do the nitrogen go?
Nitrate here.
Mhm. Yeah, so within the soil, the nitrogen is mineralized by microbes and other biological processes that convert it to nitrate, nitrite, and ammonia. So the couple forms of nitrogen. And the nitrate, it can get confusing. The nitrate is what becomes plant available. So some of that nitrogen is inevitably lost to other forms of nitrogen not available to the plants.
I'm asking about the nitrogen within the plants.
The nitrogen within the plants. Okay.
So your plants are actually uptaking nitrogen early in the season,
Mhm.
right? At the end of the season, you're seeing this low nitrate level within the plants.
Mhm.
Where does the other nitrate go?
The nitrate that we're observing from the beginning of the season.
Yeah.
So I would say that goes into biomass, because the nitrate that is in the plant at 30 days here is being converted. So as we're seeing a nitrate drop, we're seeing a biomass increase, and that nitrate directly transfers to like biomass accumulation as crop matures. Did that answer your question?
Yes.
So, yeah, I have a question.
Maxwell?
Great talk, Owen. It was
Thank you.
like that. My question is about maybe I didn't hear or see it clearly. During the plant health graphs you showed,
Mhm.
2020 or the first year had more lushy. Yeah, go back one.
Right there.
No, the
other
previous
one.
mistake. Again, but yeah, so
Mhm.
this one, 2021 had more lushy greens compared to 2022. But then
Yeah.
when you were talking about diseases or plant health status, you said 2021 had more, was more sickly. So I wasn't pretty sure how, maybe just visually, 2022 seemed like the year with less, I don't know, compared to the two slides after this that showed 2021.
Mhm.
Yeah, this one, that showed
Mhm.
2021 having more bad health in quotes.
So in 2021, just to kind of clarify, so you're asking why we would see a more lushy green in a more diseased or less healthy crop?
Yeah.
Yeah, so that's a great question. I wondered about that myself. And I wonder if it goes back to precipitation. In 2021, we had like a deluge over right before harvest. And I am guessing that fresh weight is diluted essentially by water within the crop. And it's something that we couldn't necessarily, in hindsight, dry matter would have been a good measure to take. But that's my guess, but I can't explain why it's half in 2022 versus 2021.
There's no way Owen would have known this. It came up at a student master's proposal talk yesterday, because we were growing carrots on the same plot. The site in 2022 was highly compacted, weirdly compacted. So now that I wonder if there was something to do with root growth and compaction. There's no way you would have known that, but we thought because of the carrot roots were very, very stunted. So there could have been a difference within the site. I don't know what caused that degree of compaction.
And that's why I wanted to show the visual, because I have a lot of pictures. I'm like, I'm seeing this visually, I'm seeing it in the data. Yeah, Amanda?
Yeah, I just wanted to comment that in the disease piece, and you and I worked on this, and with celery, one of the diseases that is seed-borne, and it's very odd. It's unlike other Caltrophoros and Anthracnose.
Mm-hmm.
It's seed-borne in celery, and it looks bizarre, right? It creates like a heart rot that looks like a calcium deficiency, and then things start to gnarl and twist up, and then it's like this central heart rot.
Mm-hmm.
But it's just a strange disease, and it doesn't progress with sort of consistency. It's not, its incidence in a seed lot you wouldn't know, right? So the appearance of this particular disease is just odd, right? And it's a relatively new disease to see it in commercial celery. And that's where I've worked with it in conventional commercial celery. But so that piece I think was really hard, because not knowing what's in the seed, not knowing what's in the transplants. And then so you're getting kind of this unknown that you take to the field and plant out. And there may have been some, you could get differential pathogen load in both years, right? So it was a real challenge to try to measure with consistency the disease. And that's why you and I talked about like maybe just do a health rating, because there's weird stuff going on. And it takes diagnosis of almost every plant you see it in, because it's looking so different. And so it was really a complication in this celery work.
And that's a really important point, Amanda, because one thing I left out in my methods for 2022, we did a heat bath treatment on the seeds. So the seed source was the same, and we confirmed anthracnose in year 1. Like, how can we be proactive about this going forward? Worked with Amanda. It's a pretty simple heat treatment protocol. Submerge the seeds in heated water at a determined rate for a determined time, and then dry them out and then seed them. We didn't do that in 2021. So I'm not drawing conclusions from that, but that was a change that I didn't mention. And that would eliminate this, that would reduce the amount of seed-borne diseases, that heat treatment.
But it wouldn't account for the you wouldn't expect less biomass, something.
No, and there could have been anthracnose latent in the field. There were other apiaceous crops like carrots grown in previous rotations.
Yeah, also the previous, so when you did 2021 and you applied, say the maximum nitrogen you applied, did you do anything to reclaim, or you use another plot, or how did you go about had maybe excess nitrogen the previous year, and now you wanna randomize it for your RCBD, you still use the same site that had higher nitrogen previous year or next year, or you just use a different plot?
Yeah, just a different plot entirely. Our assumption going in, I measured soil samples over the season, and we measured the total nitrogen. So that's ammonium nitrate, nitrite. And we considered that contribution each year to be nominal over the season. And the parts per million were under 100 on average for each season. It was like in 50s and 60s parts per million. So to answer your question, we assumed no nitrogen when we entered the field.
Good.
Okay. Thank you.
Oh, and another question here from Zoom. Zachary Ray is asking if you noticed a trend where the general distribution of biomass shifted from being primarily leafy to primarily petioles, and did that coincide with the reducing nitrate concentration trends?
Yeah, that's a good question. So the first question I'll answer is, did I notice a trend from a more leafy plant to a more petiole plant? And I'll say yes. Earlier on the season, the petioles were probably the size of my pinky. And and they were a lot smaller. The canopy, the leafy part, was more, I would say, a 1-to-1 ratio with the petioles. Whereas later in the season, the petioles were very thick, more than my thumb, and the leafy material was more limited to the upper third, I would say, of the canopy, more representative of what you would find in table celery at market. And then could you remind me of the second question?
Yeah, so did that change from going from primarily leafy to primarily petiole growth? Did that coincide with the reducing nitrate concentration trend that you observed?
Yeah, so that's something that we didn't study when we juiced the petioles. We juiced the leaves and the petioles, the leaves and the petioles together as one unit. So we didn't separate the biomass ratios, and we didn't compare those to a nitrate basis.
Any other questions?
Yeah.
Right at the top of the hour.
Mhm.
Let's give Owen.
Thank you, everybody, for coming. Please finish the snack.